Hello, and welcome to Season 2 of the Canadian Epigenetics Environment and Health Research Consortium, or CEE-HRC podcast, presented by their trainees. The goal of this podcast is to chat about some of the explosive and infinitely interesting research happening in the realm of epigenetics, and particularly among the brilliant scientists here in Canada. We hope you enjoy this episode.
Today, our guest is Dr. Federico Gaiti, scientist at Princess Margaret Cancer Center, University of Toronto. So, Fede, to start with the podcast, can you tell us something about why do you like science and what's your motivation to become a scientist?
Yeah, thank you for inviting me. And yeah, so I think, one of the main motivation for me has always been curiosity. So I think, I'm genuinely curious about learning new things. And, I think science perfectly match that sort of character trait, let's say. Because you come into work and you're curious about discovering something, and at the end of the day, also try and make like an impact in whatever field of like research that or like science that you are working on. And for me now it's like biomedical. So if I can combine my curiosity to learn something new with also discovering and maybe like innovating in the biomedical space, that it combine really all what I want to do in terms of like scientific, let's say, career. Yeah, so mainly I think it's the pleasure to come into work and discovering something new and learn something that I didn't know the day before. And it could be as small as reading a paper to a major discovery to something. I think I always feel I'm learning something every day, which is what driving me basically to come to work every day.
So what was your training journey to your position today? Your PhD, your postdoc, what was it like?
Yeah, so training. So I've done my undergraduate and master at the University of Bologna in Italy, studying mainly general biology. But then as part of my master, I also trained at the University of California in Santa Barbara. And there I started taking more like a branch about marine biology, actually. And during the PhD, then I moved to the University of Queensland in Australia, where I still under the big umbrella of like marine biology, but the main question was to use like genomics approaches now to try and understand basically the origin of multicellularity. And, this was sort of very much like related to what I'm doing now here in Toronto, because, you can think about cancer, if you will, as sort of from an evolutionary like point of view, is like something goes wrong at some point and one specific cell decided to like, dissociate or like in a way behave like differently than the rest of the other cell in the ecosystem. And so that sort of create and kick off all of the cancer process. So I sort of move from the two, let's say, opposite part of what I think it is the same spectrum. But basically, try now to understand what happened when a cancer cell goes average. And in order to sort of bridge the two, I did a postdoc in New York, where I also mainly learn a lot about single cell genomics, single cell methodology, but also expanding more on basically the concept of epigenetic, which is what I'm mainly interested in.
So you worked at many places in the world, in Europe, in the US. How different is it from one place to another?
Yeah, so I mean, I think, it's sort of difficult to compare one to one, because, I was also at very different stages of my career. But, I can tell you, not only stages, but also research field. So that's also like impact. So, from my personal experience, North America was much faster pace, let's say, than what I experienced in Australia, where the environment, partly also because I wasn't in the biomedical field, it was a bit more like lawyer key and like slower pace in a way, like the community was a bit smaller because, the evolutionary marine biology community is not as big as the biomedical world. But, in terms of research environment, I think, they're all very good places. And, I enjoy all of the three continents. I guess I don't really had a major research experience in Europe. It was more like throughout my undergrad. But, I would be happily work in a scientific research institute in Australia. And I think, Canada, in general, North America is equally good. I think many other aspects, comes into play when you decide where you're going to move longer term. But I sort of think as of Canada as I'm having similar vibes and feeling when I was in Australia. I think, don't think that's pretty much
So you're describing what we are having here in Toronto is more slow pace?
It's not slow. It's definitely faster than what I was feeling in Australia in terms of day to day. But again, I also change field, so I can't really compare Apple. It's not really an apple to apple comparison here because I wasn't doing biomedical work back then. But in terms of quality of the research, they are both fantastic places, and there are some major, institute in Australia that, again, I would be happily working there, beside the fact that you are 15-hour flight away from the rest of the world. So then, it also comes down to, but, North America and Australia, for me, I place them, at a very equal level in terms of research quality and environment. So, of course, the US might offer more diversity because, there are many more research institutes. And so that might be a pros about, working in the US. And you have more opportunities, let's say, in working in cutting edge research, simply because there are many more.
So as you mentioned before, you had a switch of research from marine biology to biomedical. So I'm just curious, from studying sponge genome to human genome, have you ever encountered any challenge and how did you overcome that?
Yeah, I mean, honestly, the main challenge was to catch up and learn quickly from the transition from PhD to postdoc about cancer biology. Because, I had the background about general biology. I had the background about genomics, which, whether you're working on a sponge genome or a human genome, the principles are similar. So I think for me was really the fact of having to catch up with the whole cancer biology field, which I was never really exposed to that before, and that it took definitely months, years, and I'm still catching up and learning every day. I'm not myself a cancer expert. I consider myself more like a genomics expert than a cancer expert. And so what I'm trying throughout the year is to sort of combine these two worlds about, learning about cancer biology using genomics or epigenetics, let's say, approaches. But yeah, that was mainly the challenge. And but I'm still working through that.
So, for you established your lab last October, so we're still very new. How would you summarize your new PI life so far? I guess it's still a major transition of your career path.
Yeah, so I mean, so far I think I can say I'm very much enjoying, and it's been very nice experience. So let's say I'm enjoying the ride. Let's say it's definitely coming with more responsibilities than doing postdoc or PhD, but that sort of was expected. And I'm really enjoying actually supervising students. I haven't had the chance to teach yet, but I will do it this coming semester, and I'm excited about that too. So I think it's definitely more responsibility and a lot of different tasks that you need to be able to do and do well. But, mainly if you surround yourself by a good team of people, they will help you out throughout the way. So I think this is what I'm experiencing now. There are a lot of tasks that I had no idea how to do before, but there's always someone willing to help, and the team in the lab is actually very important for that, but also other more senior investigator, all of like the admin people. So you are not like left alone, even if you have to do many different tasks that you had no idea before, like, including, like financial issue, interviewing people, all of these things. So the main one I was exposed before was like writing fellowship and grants. Mm-hmm. And that sort of like stay alive, remain. But, all of the other aspects about more, let's say, the management side of things, at least myself, I wasn't really exposed to much during my postdoc. And all of a sudden you have this new like responsibility and like task. So, you learn as you go, and as in everything in science, I think the best way to learn it is to learn from your mistake as well. So, if it happened that you calculate something or you hire someone that is not as good as you thought, at least you have learned something.
As a new PI, I expect you to have a very busy life, but what do you do in your spare time, and how do you balance your work and life?
Yeah, so to be honest, I'm definitely try to balance my work and personal life. I think you need to be able to not just work like 24/7, but really take time to like refresh. So, I like running, so I run a lot, bike a lot, and mainly the weekend I go for long run, long bike ride, and I like cooking. So myself and my partner, we like cook a lot, experimenting back home as well, like making pizza and try various different flour batches and things. So that's also something fun. I keep sort of the fun of doing science, but in a more like relaxed home environment. Mm-hmm. And mainly, and then take also my time to see family. So I usually, go back home and see family or like friend, and do some like small trip, and mainly this. But I always try, and there are the week or like period that are more intense than other. But when I leave the office, and I'm not going back home and work more. So, I always try to like detach from what I'm doing like in the office, and I need my space. So I actually think it's not the most productive way of dealing with science if you work just 24/7. Mm-hmm. It's not the most efficient way to do it.
Yeah. And where is your best spot for running in Toronto? Are there any good places?
I mean, I'm lucky enough that I live very close to the lake. So I just really love the path along the lake. And you can go both way. I try both, either going like west or east. East you get to the beaches, which is like beautiful. West you can run as much as you want, like Sissaga and onwards. And so it's very nice. I like along the lake. Give me a sense of peace and relax when I run along the lake.
Yeah, I understand that.
So I think it would be a nice ending for part 1. Let's talk about some hardcore science. So Fede, can you share with us what is your definition for epigenetics? How do you understand it?
Yeah, so epigenetic, so I think that term of epigenetic, there's still a bit of like debate how people define that. But like for me, I interpret it as probably the original way, which is more like involving like heritable mechanism that are not attributable basically to changes in the DNA sequence, but somehow they are affecting cell fate or cell state. And this is sort of what I think about like epigenetic is, and basically like describing more like properties that involve cell state and cell fate that are not directly changing the DNA sequence. And this should be like heritable mechanism. And then, of course, you can also maybe like interpret that more in like a modern way, let's say, or like more like as a genomics or molecular biology perspective, which is basically all of the layer of regulation that happen beside basically changes in the DNA. And, that doesn't really comprehend or like heritable mechanism. So I tend to stick more to the original version of there need to be some heritability when we are talking about epigenetics.
Which aspect of epigenetics are we studying in your lab?
Yeah, so we've been curious about mainly DNA methylation, and this is sort of like a continuation from what I was doing during my postdoc in New York, and mainly also in the context of brain tumor, where we know that DNA methylation is really important. And we are also expanding to other type of epigenetic, if you want to thinking about in the context of like a layer of regulation. We are expanding now to also looking more at like chromatin, but DNA methylation is still the main layer that we are interested in.
I have a question for that. So you worked in sponges and now you work in human cells or mouse. How conserved are the enzymes and everything that regulates the methylation of DNA across species?
So I think, I don't want to say, so I haven't really done the specific analysis about the enzyme involving methylation, but there is a group in London that actually did it. And I think they're pretty conserved across basically animal. The thing I remember is that the sponge genome is very hypermethylated for some reason. I think they probably described that in that paper. And the paper is actually from Alexander de Mendoza. So if you're curious, but from my PhD, I was more looking at histone modification, and those I can tell you for sure that they are indeed conserved.
Oh.
And I think methylation, they found some conserved pattern. There were some differences as well that I can recall now, but the sponge genome is definitely hypermethylated. Don't know, definitely interesting.
Tell us something more about your research on glioma. What was the background story? What originally attracted you to this specific disease?
Yeah, so I think we were interested in that because, we know that in cancer there are certain cellular phenotypes, such as, they can be like persistence or plasticity that, they need to be inherited to contribute to actually cancer evolution. But often these are sort of like uncoupled from actually genetic identity. And one case is actually human gliomas. So in this tumor, they've been finding a transcriptional heterogeneity that, were only partly explained by genetic changes. And so for us, it was really the motivation of like, well, we know that these tumors are very diverse, and we know that it's not explained fully by genetic changes. So there must be something happening at the level of the epigenome. And so we went ahead and basically asked a very simple question about, what are the epigenetic mechanisms that underlie this diversity in this brain tumor cell. And we started with methylation because it's usually considered the most heritable mark. And we also had the right technique to probe specifically methylation at the single cell level. That's how we got into methylation and in brain tumor.
Since you're doing it in single cell, what are the benefits from doing that in single cells compared to bulk?
Yeah, so the benefit is really that you can get an understanding about this diverse cellular subpopulation that you will not be able to directly assess if you do like bulk sequencing. So now with all of these new single cell approaches, you are able to actually dissect this intratumor heterogeneity and start asking questions about whether there are specific set of cells within the tumor that behave differently and what are the factors that are driving this specific subpopulation. And, that can be in many contexts, can be, in the context of like therapy resistance or like recurrence, and so that's sort of the overarching question that we are interested in, like how can we probe this cellular intratumor heterogeneity further, not just at the level of transcription, but also other layer of the epigenome.
And do you think a lot of the variability is caused by the changes in DNA methylation or in histone marks or other epigenetic changes?
I mean, that's a great question. I don't know if we have evidence. We can say what, I don't know if we have evidence about causation. What we can say, at least in the study that we published using DNA methylation, we definitely provided evidence that at least DNA methylation changes reflect this brain tumor cellular state and may contribute to their propagation. Basically, that's pretty much as far as maybe I can go with claiming what caused what. We will need to do more like mechanicistic work, and we are not there yet, at least in the lab, but other people are working on this. But it's an interesting question, and definitely what came first, basically. What caused what.
Can you give us a description of what were your major findings during your postdoc research on glioma and single cell methylation?
Yeah, so I think the main finding was that, they've been previously shown, again, that these like glioma tumor are diverse transcriptionally. And so the major finding was that actually we had the ability to now ask what were the difference at the level of DNA methylation. So we found a set of cells which were the less differentiated cell, let's say the cell that resemble more like stem cell, let's say, that have this particular hypomethylation of this specific complex, which is called polycomb repressive complex 2. And, this was also shown like before to being important in like regulate basically stemness. And then, so that was one of the findings. The other one was actually the fact that during my postdoc, I work on this approach using DNA methylation changes as a way to reconstruct basically tumor phylogenies. And then again, this is basically a powerful way to like reconstruct lineages, because you can do it directly from a patient sample. And so that's what we did in brain tumor as well. And one of the major findings between the two subtypes of glioma brain tumor, one that doesn't have the IDH mutation and one that does have the IDH mutation, was that the IDH wild type, we found it to be much more plastic in terms of basically cellular state, which means that over time we think that these tumors have basically the ability to replenish their stem cell compartment. And if you're thinking about it more broadly, many therapy currently are like focused on trying and deprive the tumor of their stem-like cell compartment, because that's what like canonical is what the cell that they contribute the most to tumor progression and initiation. But so if the tumor is very plastic, we think they have the ability to actually reconstitute basically this stem-like cell compartment. And so we need to come up with like different way to think about approaching in terms of like clinical perspective about this tumor, that don't follow the more classical hierarchical structure where at the apex you have the stem cell that then give rise to all of the more differentiated isolated cells. So that was the major difference. We are trying ways now in the lab as well here to dig more into this finding and assess this sort of like plasticity that we have seen in the IDH wild type glioma.
Tell us something more about the ongoing projects in the lab. What are we working on? What questions we're trying to address?
Yeah, so as a continuation, we are trying to basically expand this like finding that were published back in the fall to now expand more, looking at like spatial and also like temporal aspects. So what we published was basically a snapshot in time of this tumor, but now we want to do more like longitudinal work to try and understand how this tumor progress, and then again how plasticity affect this tumor progression, and what are the factors, especially in terms of like mutilation and underlying transcription factor that like regulate this plasticity. And also in terms of spatial. So, we know that tumor cells are not working in isolation. They work with their surrounding environment. And especially in brain tumor now have been, more and more, we know like more and more that this is actually what's happening. And so we are trying and understand better the spatial component and the contribution of non-malignant cell that surround the tumor cell, and see how they interact with each other and how they potentially contribute to this plasticity. And also expanding to other type of brain tumor, not just glioma, and try to also come up with a way to construct lineages in vitro or in vivo, so that we can apply some perturbation and try to understand how this plasticity is affected by, let's say, current standard therapy or inhibitor or some epigenetic drug. So these are the main new direction.
So you talked about the spatial aspect of the cancer. Are the cells that surround the tumors, are they going to vary a lot depending on where in the brain the tumor is? Or is the microenvironment in the brain mostly constant throughout the brain?
Yeah, that's a good question. I don't know if there were many studies comparing different locations across. Usually you have access to, where the brain tumor was, but you don't have access to other area. But I think there are now been studies where people will start to assess many different locations. I think there's definitely a gradient and in different cell type involved as you move from more the core center of the tumor to the periphery and then farther away. Whether this composition change between, within the brain or in another area, I don't think has been studied a lot, because again, I think you have access mainly to the area that is surrounding the tumor itself. But there's definitely a relationship between the type of cell and the distribution of the cell type that you see and the location. And this, many papers are coming out recently, and this is a very active actual field now.
Yeah, and you also talked about where the cell is within the tumor. So when you do a single cell sequencing of a tumor, can you know where the cell was? So if it was a cell that was most within the tumor or at the periphery, is there a direct way to see that? Or is the signature really different enough that it's easy to know where the cell was?
So I think if you don't do any pre-enrichment, or if you don't know where your cell is coming from, let's say you take your entire tumor and you sequence it, it's going to be difficult to know where your single cell are coming from. But you can pair that with images. And so then you can try maybe reconstruct where you collected the specific tumor sample, or a biopsy within the tumor, and try to then correlate with specific signature that you see in your single cell data. Or you might as well just do a spatial analysis where you actually do spatial single-cell transcriptomic, and that way you are sure that the signature you are seeing is coming specifically in that specific area of the tumor.
Yeah, it makes sense.
So that would be basically the new way, and recently the spatial transcriptomic field has exploded, and interestingly also very recently expanding to the epigenetics. So there was a very recent preprint last week or the week before about actually probing chromatin changes now and spatial. So that's very interesting.
Indeed.
So I know you're running a basic science lab majorly, but looking ahead, in your perspective, how would your work have the chance to change the prognosis or the treatment paradigm of glioma patients?
Yeah, I think that's an interesting question for sure. I think besides providing basic knowledge of the system, which is actually very important, especially in the context of brain tumor, where I still think we don't really know enough about how, the basic biology of this system and how they interact and how they evolve also with other cell type. And so I think that would be the major contribution, try to provide and increase knowledge about how this system works, with the hope to find some specific pathway or gene or signature that at least provides some guidance for more clinical people to then try but at the moment I think it's mainly directed to increase the basic knowledge about the biology of this tumor, and because that's how basically I see that you will contribute the most then in the longer term about clinical aspect. It's we still don't know a lot how this tumor behaves. So why don't we ask the question about, let's find how they behave a bit better, and then maybe we can come up with a way to address. So one example is the fact that we found this increased plasticity, especially in IDH wild-type glioma. And so, if you think about from a therapeutic perspective, you might want to start thinking about ways instead of focusing your clinical energy into trying and deprive the tumor of this stem-like cell. If the tumor has the ability to rebuild constantly this stem-like cell, we need to come up with a different way to think about this, and this can be basically targeting specific other type of cell within the tumor, or disrupt the tumor with the ability to replenish the stem cell compartment. And this will need some sort of rethinking about how we target this tumor. But first we need to understand a bit better how this plasticity actually work, and what are the factors that mainly contribute to that. And so that takes several decade I can really predict.
Mm-hmm.
But it will take a lot of effort before being able to move this conceptual idea and preliminary result to the clinic. But
I had a comment for that. Yeah, well, I think it makes a lot of sense what you just said, because if you stop the tumor's ability to evolve, then all the therapies that you're going to use against it are probably going to be more effective, because there's less chances that it can adapt to it, to resist it and come back after the treatment. So it would be a smart way to target the tumor in a long-term
exactly
treatment.
Other tumor type. So my first part of the postdoc actually was I was working on chronic lymphocytic leukemias, and I got also very interesting the concept of early cancer detection. And so the other branch that we are taking now is to try and understand better these pre-malignant stages of cancer, specifically using stages that are happening before lymphomas. So this could be individuals that have some of autoimmune diseases, or some patient or individual that they don't have yet an aggressive lymphomas or leukemia, but they are basically predisposed. They are more at higher risk and then let's say to develop this in the future. And so that's a whole area that we are interested in, and we are starting to work also in that. And that would shift the concept basically of what we are trying to do in glioma, which is finding a cure, let's say, compared to an early detection prevention approach. Not just like looking at already when the cancer is formed, but like rather when the cancer is not even initiated, and try to understand how we can even like prevent and detect early on before the cancer appears.
For the last part, we want to talk something about mentorship. Mentoring students, postdocs, it's a new major role you take on as a PI. So to start with yourself, during your whole scientific training process, is there a mentor that stands out that give you a big impact on your career path?
Yeah, I mean, I'd say, at every step, I was lucky enough to have met basically mentor that I think they very much contributed to increase not only like my knowledge, but like fuel my curiosity. So the first one was definitely my undergrad research advisor in Italy, and yeah, a guy super passionate, full of energy, and it was just so much fun and nice to work with him, and I learned a lot, and I saw how like passionate he was to go to work like every day, and sort of that stick to me is still like these days. PhD advisor and also co-supervisor were amazing people and friends too, and they had a big influence. They guided me through all my PhD, and yeah, I learned a lot from them, not just in terms of like science, but also how to deal with daily up and down and like challenges that you constantly face, especially as a PhD student. And yeah, postdoc, I learned a lot in terms of working hard, and I'm learning how to gather high impact publications, scientific writing, grantsmanship, all of those like aspects that, were more like relevant to my current stage of the career now. And yeah, so they were all really, I would say each mentor at each of the steps of my career was really a great person and great scientific advisor and a great mentor. So I can't really complain. I feel lucky about the mentor I had.
Now, as a mentor yourself and actively recruiting new trainees in the lab, how would you describe your own mentorship style?
Yeah, I think my mentorship is to give freedom to the people to express themselves and follow what they are curious about. Of course, falling under, the main interest and approaches that we are taking in the lab. And, I'm always for people that are like passionate and curious, which again, I think are probably the main two features that I like about like science. So I'm looking for those type of like traits when I deal like my team. And so mentorship, is just to try to have a sort of like an open communication with my team and with the student, and like guide them through, but also make them learn through the mistakes. And because that's how I did it, and I think is important to realize that science is not successful all the time. I think it's actually the contrary. Is most of the times you deal with like micro failure, and these are not failure that it's not that you are failing, but the experiment might fail, the code might have a bug, and you find something that it doesn't work, a result might not be the one that you expect. So these are all like micro challenges on a daily basis that you need to deal with, and that that how I think you learn, because you learn from your mistake, but you learn from failure, you learn from a negative result. And so, it's that. So that brings me to also the other trait that I value a lot, which is persistence. So I think that, to be a good scientist, you not only need to be curious and passionate, but also have some level of persistence. Being able to, in a way, don't fall at challenges, but take that as a learning exercise and don't give up and try again. That's sort of how I feel. I've been doing this basically, throughout the undergrad, PhD, postdoc, and now here. It's a constant learning process, and you make mistakes, things don't always work, but you learn, you try again, and you move on, and you learn more, and you learn more, and I don't think there is an end on how much you can learn. So, it's a nice ride.
So, working in the lab, we're really seeking for your guidance and mentorship. But at the same time, one major goal of graduate study is to develop independence, to be able to do science in the future without you. So regarding that, what would be your suggestion to trainees, how to gradually develop independence during this whole PhD process?
I think it would be really to take some risk in the way that, being like proactive about coming up with like idea, don't be afraid to fail. And, that like slowly, and that of course you need to have also like a mentor and a supervisor, which is open, to a new idea and like this sort of like discussion. It's not all like predefined boxes, but, if you like proactively start training yourself about stepping out a little bit about your comfort zone, asking new questions and thinking about new idea, and not just with your supervisor, but, even with your peer or other people, then you are constantly basically thinking about new idea, and you bounce back this with people, and, like slowly you develop this sort of like level of independent like thinking that you don't need any more people to then tell you today you are gonna do this. You proactively coming up with the way to like address a question that you are interested in. But to do that you need to step out a little bit from your comfort zone, because you might feel a bit like unknown, like thinking about something that you don't know, or like a new idea, and you have a lot of questions around that you have probably never thought about it before, and that can make you feel a bit like uncomfortable, but that's part of the learning and like growing process. So I think, if you keep asking yourself questions and being curious, that's already part of developing your independent thinking, basically.
Yeah, it's a really nice vision on how to grow in science.
Yeah, and hopefully it is. Maybe I'm a naive new PI, but medicine. But that's how I think. I keep thinking about, and definitely though you need to be willing to step out of your comfort zone, and like take some risk, and be also at peace with yourself in a way that if you step out of your comfort zone, failure or mistake might be behind the corner, but so that's the infinite loop of like learning process. You just
Yeah, and it's good in science, but it's good for everyday life too. It's just an advice that fits for everything.
Yeah, that's how indeed where I think how I generally approach things, not just in science. That's how I do things. I like to, like stepping out a bit of my comfort zone and try new challenge, and even like simply like running at a few more K a day, or running a bit faster, see if I do that. If not, learning why I wasn't able to do that. But like even like in personal life, I think just need to be able to like react to basically more the negative results and learn from that. You grow that way.
I think it would be a nice ending for the podcast. So thank you so much for being with us today, and it was a great talk with you.
Yeah.
Thank you, guys. Yeah, I really appreciate it.
Thank you for listening to Season 2 of the This is Epigenetics podcast. If you like this podcast episode, you can check out our website and future episodes at thisisepigenetics.ca. We hope that you all have a very good day.